Hello to all dear students. In this video, we are going to discuss about a lipid derived autocoid, that is eicosanoids, and we are basically emphasizing upon prostaglandins, thromboxanes and leukotrienes. So first of all, about eicosanoids. The eicosanoids are oxygenation products of polyunsaturated long chain fatty acid. The thing you should emphasize over here is these are oxygenation products of poly. Poly means many, and what is many? Unsaturation means there are many double bonds over here. And basically this icosa means compound containing 20 carbon atoms. Next, talking about two important examples of eicosanoids, that is prostaglandins and leukotrienes. Prostaglandins and leukotrienes are biologically active derivatives of 20 carbon atom polyunsaturated essential fatty acids that are released from cell membrane phospholipids. They are the major lipid derived autocoids. Now talking about arachidonic acid, which is the precursor for the synthesis of the eicosanoid, that is prostaglandin and leukotrienes. Arachidonic acid, abbreviated as AA or 5,8,11,14 icosa tetraenoic acid, the most abundant of the eicosanoid precursors, is a 20 carbon atom fatty acid containing 4 double bonds. So we'll check out the structure of arachidonic acid. First of all, this is COOH. So here you'll find one carbon atom. Next, carbon atom number 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 and 20. So this is icosa. Icosa means compound containing 20 carbon atoms. Next, what else we require? We require tetra double bond. Tetra double bond means 4 double bonds, and the position of these double bonds are at 5, 8, 11 and 14. We have got double bonds. Okay, so this is icosa, that means 20 carbon atom. Tetra means 4 double bonds, enoic acid for COOH. So this is arachidonic acid, also known as 5,8,11,14 icosa tetraenoic acid. Arachidonic acid must first be released or mobilized from the SN2 position of membrane phospholipids by one or more lipases of phospholipase A2 type for eicosanoid synthesis to occur. So suppose this is your membrane, or you can say plasma membrane, which is mainly composed of phospholipids. So basically this arachidonic acid is esterified in membrane phospholipid and we need to mobilize it. So stimulus such as diverse physical, chemical, inflammatory and mitogenic stimuli, it will cause activation of the enzyme phospholipase A2. Next, free radicals gets converted into isoprostanes, causing the release of the arachidonic acid. Now once this arachidonic acid is been released, it can be acted upon by two enzymes. First one is cyclooxygenase enzyme and second one is lipooxygenase enzyme. When cyclooxygenase enzyme acts on arachidonic acid, it causes the production of prostanoids such as prostaglandins, prostacyclins and thromboxanes, whereas lipooxygenase pathway will lead to the production of leukotrienes. So let us start our discussion with the cyclooxygenase pathway. So arachidonic acid is acted upon by the enzyme cyclooxygenase. This enzyme is abbreviated as COX, that is COX, and we have two types of COX, that is COX1 and COX2. So now first let us know something about these two varieties of COX enzyme, and then we shall resume back to the cyclooxygenase pathway. Okay, so cyclooxygenase enzyme are of two types, COX1 and COX2. COX1 also known as prostaglandin H synthase 1. COX2 also known as prostaglandin H synthase 2. COX-1 expressed constitutively in most cells. It is synthesized and is active in the basal state. The level of COX-1 activity is not much changed once the cell is fully grown. Whereas COX-2 it is inducible by cytokines, growth factors and other stimuli during the inflammatory response. So by this we can know that COX-1 is involved in physiological activity, whereas COX-2 is active under pathological conditions. Next, it is believed that eicosanoids produced by COX-1 participate in physiological, that is housekeeping functions such as secretion of mucus for protection of gastric mucosa, hemostasis and maintenance of renal function, while those produced by COX-2 lead to inflammatory and other pathological changes. Now coming back to our cyclooxygenase pathway. Upon the arachidonic acid, both COX-1 and COX-2, it will promote the uptake of two molecules of oxygen by cyclization of arachidonic acid to yield C9C11 endoperoxide C15 hydroperoxide. This product is known as PGG2. Next, this PGG2 it is rapidly modified by peroxidase moiety of the COX enzyme to add a 15 hydroxyl group that is essential for their biological activity. This compound is known as PGH2. Now we will revise this thing. Arachidonic acid first get converted into PGG2. This PGG2 is rapidly converted into PGH2. These both PGG2 as well as PGH2 are unstable endoperoxides. This PGH2 it can move to various body tissues, and in presence of enzyme isomerase it can form either PGE2 or PGD2 or PGF2 alpha. Next, PGH2 when it moves to the platelets, in the platelets it will found the enzyme thromboxane synthase and it will lead to formation of TXA2, which is thromboxane A2. Next, PGH2 it can move to the vascular endothelium where it will find the enzyme prostacyclin synthase to form a compound known as PGI2, that is prostacyclin I2. Now, if you carefully see all these structures, PGE2, PGD2, PGF2 alpha, thromboxane A2, PGI2 and even PGH2, what thing you will find common in all these structures? Yes, in these structures you will find that all have got just two double bonds. Okay, everywhere you can find two double bonds. So this is common. And another thing that is common in all these compounds are all have got a subscript of 2. So this subscript 2 mainly denotes the number of double bond. So all the products of cyclooxygenase pathway have got just two double bonds. Now you just revise from where we started. We started from arachidonic acid. Arachidonic acid is a compound containing 20 carbon atom and 4 double bonds. But in cyclooxygenase pathway what happens? 2 double bonds are being utilized for the formation of cyclic ring and only 2 double bonds will be remaining intact. So in cyclooxygenase pathway all the products will have just 2 double bonds and therefore they have got the subscript of 2. And moreover, these all the compounds whatever you can see, these are synthesized endogenously, means inside our body. We can also synthesize these prostaglandins in the laboratory. Like for example, we have got misoprostol and alprostadil. These two compounds are an example of PGE1. PGE1 means the structure will be same as that of PGE2, but instead of 2 double bond it has only 1 double bond. Okay, whatever may be the subscript, that many number of double bond will be there. Okay, like in PGE2 we have got 2 double bond. In misoprostol and alprostadil, these all are the examples of PGE1, means it have got just 1 double bond, but it functions same as that of PGE2. Next, if you see carefully, this PGE2, PGD2 and PGF2 alpha. These three compounds are collectively known as prostaglandins. So basically, prostaglandins have a 5-membered ring with 2 side chains projecting in opposite directions at right angle to the plane of the ring. Next, this TXA2 is thromboxane and this PGI2 is prostacyclin. Now, the prostaglandins, thromboxane and prostacyclins, these three compounds are collectively termed as prostanoids. And how these prostanoids are synthesized means from the downstream, starting from arachidonic acid, which is first converted into PGG2. PGG2 is rapidly converted into PGH2. Both this PGG2 and PGH2 are unstable cyclic peroxide, and this is rapidly converted into prostanoid by the enzyme isomerase. Next, about this PGE2 and PGF2 alpha, these both are primary prostaglandin and names are based on the separation procedure. Like PGE, it is mainly partitioned in ether. So E for ether, that's why the name PGE2. 2 is for the 2 double bond and E is given because it is partitioned in ether. Similarly, PGF2 alpha is partitioned in phosphate, but in Swedish phosphate spelling is FOSFAT. So we are giving PGF2 and alpha. Alpha in PGF2 alpha refers to orientation of OH group on the ring. Next, platelets primarily synthesized. These platelets primarily synthesize thromboxane A2, which is chemically unstable, spontaneously changes to thromboxane B2. Even this endothelium mainly generates prostacyclin, that is PGI2, which is also chemically unstable and rapidly converts to 6-keto PGF1 alpha. Okay, so this is all about cyclooxygenase pathway. Next, we will check out the lipooxygenase pathway. So this pathway appears to operate in lungs, white blood cells and platelets. Upon the arachidonic acid acts the enzyme lipooxygenase, that is 5-lipooxygenase, in presence of a protein known as FLAP protein. FLAP means 5-LOX activating protein. This enzyme plus this protein both acts on the arachidonic acid that results in production of a compound, that is 5-hydroperoxy eicosatetraenoic acid, abbreviated as 5-HPETE. Now this compound is rapidly gets converted to an unstable epoxide leukocyte known as leukotriene A4, abbreviated as LTA4. Next, this intermediate either gets converted to dihydroxyleukotriene, that is LTB4, by the enzyme LTA4 hydrolase, or it gets conjugated with the glutathione to yield leukotriene C4, that is LTC4, which undergoes sequential degeneration of glutathione moiety by peptide to yield LDD4 and LTE4. Now here also you should check out the subscript is given 4, and this 4 denotes the number of double bond. As you can see anywhere, LTA4, LTB4, LTC4, LTD4 and LTE4 all have got 4 double bonds. Therefore the subscript of 4 is given to it. Next, the 3 last products, that is these LTC4, LTD4 and LTE4, these 3 products are collectively known as cystinyl leukotrienes. Next, about this LTC4 and LTD4. LTC4 and LTD4 are potent bronchoconstrictor and are recognized as the primary component of the slow reacting substance for anaphylaxis, that is secreted in asthma and anaphylaxis. So this is about the synthesis. Next, actions and pathophysiological roles we'll check out in my next video. I hope you people found this video helpful, and thank you for watching.